Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize-winning lab. This channel has three main goals. One, to help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. Two, to build a supportive community where we can exchange the most valuable experience-based research advice. Three, to share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the third goal. Let's go! Let's make great science together.
Welcome back to the Deep Dive. Today we're tackling something really fundamental, how tiny machines inside our cells actually work. Specifically, the protein shredder, ClpXP.
Yeah, and it really turns some old ideas on their head.
Totally. You know, we often think of these molecular motors like these perfect little engines, right? Like clockwork, sequential steps.
Mm-hmm. The standard rotary model.
Exactly. But this research we're diving into today, it shows ClpXP is actually both stochastic, kind of random in its first move, and somehow still highly coordinated.
And here's the really counterintuitive bit right up front. This tiny shredder, it can take these huge 4-nanometer steps.
Huge on a molecular scale, yeah.
Right. Even if only 2 of its 6 sort of fuel injectors, the parts that use ATP energy are actually working.
That just sounds wrong, doesn't it? How can you get such a big result from a crippled machine?
Well, that's the puzzle this paper solves. Our mission today is to unpack stochastic but highly coordinated protein unfolding and translocation by the ClpXP proteolytic machine. It uses some amazing single-molecule tools to, really settle some long-standing debates about these AAA+ machines.
Okay, so maybe just a quick refresher first. AAA+ proteins, what's their main job again?
They're basically the cell's quality control garbage disposal system for proteins. ClpXP is a classic example. It's got 2 main parts.
Right, the ClpX part, which is this ring of 6 subunits. The motor uses ATP for fuel.
Mm-hmm. And then there's the ClpP part, which is like a little chamber, a barrel, where the protein actually gets chopped up.
So ClpX finds a protein that's been tagged for destruction,
Mm-hmm.
usually with something like an S or A tag.
Yeah, deGrom tag. It grabs onto that tag, then mechanically unfolds the protein, pulls it straight, and then spools it, translocates it into the ClpP chamber.
And the big question for ages was about those 6 subunits in the ClpX ring. Do they fire one by one in order, like 1, 2, 3, 4, 5, 6, bang, bang, bang?
Or is it more random, like subunit 3 fires, then maybe 5, then 1? Old methods couldn't tell. They just averaged everything together. You couldn't see individual steps or if there was any pattern, any memory in the motor.
So how did they see it then? This sounds incredibly small and fast.
Well, that's the methodological leap, right? They used single-molecule optical trapping. You might know it as optical tweezers.
Okay, tweezers made of light, basically,
Okay.
to hold on to single molecules.
Exactly. It's a huge challenge because you're trying to watch steps that are just, 1 to 4 nanometers. Tiny.
Yeah.
In real time.
So what's the setup look like?
Picture this. You've got the ClpXP motor stuck to one tiny bead held by a laser trap.
Okay.
Then you have the protein. It's going to shred, say, a few copies of a protein domain called Titan, linked to that SSRA tag attached to a second bead held in another laser trap, and there's a little DNA linker connecting them.
Got it. So you can measure the distance between the beads really precisely.
Precisely. And you watch what happens. When ClpXP grabs on and starts pulling, suddenly you see the distance jump up.
Because the folded protein domain gets yanked straight.
Right.
Unfolded, it takes up more lengths.
Exactly. That's the unfolding event. A sharp jump. And then right after that, you see these smooth downward steps.
That's the motor pulling the unfolded chain in.
Yeah.
The translocation.
Yep. Spooling it into the destruction chamber. You can literally watch the shredder work step by step. It's beautiful data.
And they use different versions of that Titan protein.
Right. They use the normal wild type, WT, and then 2 mute versions, V15P and V13P. These mutations subtly change how stable the protein is, right near where ClpXP grabs on.
Okay, so they're testing how much the protein's own structure resists the pulling force. Does it fight back?
That's the question. How does that local stability affect things?
So let's talk about that initial fight, the unfolding part. They measured something called the pre-unfolding dwell time.
Yeah, basically, how long does ClpXP have to pull and strain on that folded domain before it finally pops open? It's a measure of the protein's toughness, its resistance.
What do they find?
A really clear result. The unfolding time was directly linked to how stable that bit of the protein was. If it was stable right where the motor was pulling, the motor really struggled.
How much difference are we talking?
Big difference. The least stable one, V13P, popped open pretty fast, around 5.9 seconds on average.
Yeah.
The middle one, V15P, took longer, maybe 17 seconds.
All right, quite a bit slower.
But the really stable one, the wild type Titan, it held on for at least 55 seconds, maybe longer. That was just the lower limit they could measure reliably.
Wow, 55 seconds. That's ancient on a molecular timescale.
It really is. So it confirms, that the protein's own structure right near that tag is the main thing slowing the enzyme down initially. The protein's stability dictates the unfolding speed.
So it's not just about the motor's power, it's also about the substrate's resistance.
Absolutely.
Yeah.
It tells you if you want a protein to last longer in the cell, maybe you just need to engineer that spot near the tag to be really mechanically tough.
Huh. Interesting. And did they see anything about how it unfolds? Like, is it one big struggle or lots of little ones?
Mostly it looked like one main hurdle. The dwell time data mostly fit a single exponential decay, suggesting one primary rate-limiting step to pop it open.
Okay, so once it gets past that hurdle, it's unfolded, then what? Translocation.
Right, and then it shifts gears into spooling the unfolded chain. And here the speed is pretty steady, actually. Around 24 amino acids per second for those Titan bits. The actual sequence of the protein chain has a bit of an effect, but not huge.
Did it ever, pause during the spooling?
Yeah, it did pause sometimes. And those pauses seem to depend a bit on the amino acid sequence going through. Maybe the chain briefly folds up into some little structure that makes the motor hesitate. That's the idea.
But the really cool part was looking at the individual steps during that spooling, right?
Wow.
In Figure 4.
Oh, yeah. This is where they saw what the averaging in the old experiments hid. The steps weren't all the same size. They saw clear steps of 1 nanometer, 2 nanometers, 3 nanometers, and even 4 nanometers.
Whoa. So multiples of about 1 nanometer.
Exactly. And crucially, the pattern was definitely not sequential. It wasn't just 1, 1, 1, 1 or 2, 2, 2, 2. They saw traces like, 1, 2, 1, 1, 3, 1, 4, 2, all mixed up.
So that just completely rules out the simple clockwork rotary engine model, doesn't it?
It really does. The firing order of the subunits seems probabilistic stochastic.
Didn't you say it was also coordinated? How can it be random and coordinated?
That's the surprising twist. They found evidence of enzymatic memory.
Memory. What does that mean for a motor?
It means what it does now influences what it does next. It wasn't pure chaos. For example, if it took a 1 nanometer step, it was actually more likely to take another 1 nanometer step right after.
Okay.
And similarly, if it took a longer step, 2, 3, or 4 nanometers, it had a tendency to follow up with another long step.
Huh. So it's like it gets into a gear, a low gear with small steps or a high gear with big steps, and it kind of stays there for a bit.
That's a great analogy, yes. It suggests the overall state of the 6 subunits, which ones have ATP, which have ADP, which are empty, that state persists for a short time, and biases the next step. It's not a rigid clock, but it's not totally random either. It has preferred modes of running.
And that explains why sometimes the whole process is faster or slower overall, because the machine can get stuck in these different gears.
Exactly. You see different populations of motors running at different average speeds.
Okay, this leads us to the coordination part and that really wild experiment with the hobbled motor.
Yes. Figure 5. This is maybe the most mind-bending part. They wanted to know how many subunits need to burn ATP to get those big steps. Does a 4 nanometer step absolutely require 4 ATPs to be hydrolyzed one after the other?
Seems logical, right? 1 ATP per step, roughly.
That was the assumption. So they engineered a mutant ClpX ring. They called it RWE.
RWE. What does that mean?
It means the 6 subunits in the ring alternated. RWE, RWE. The W subunits were normal, wild type. They could burn ATP. But the R and E subunits had mutations that basically broke their ability to use ATP effectively. They were like, duds. Placeholders.
So in the whole ring of 6, only 2 subunits could actually provide the power stroke from burning ATP.
Precisely. A severely hobbled motor. And it was weaker. It unfolded things much slower, like 8 times slower, and couldn't pull as hard, only up to about 10 piconewtons of force compared to 26 for the normal one.
Okay, so it's definitely impaired. Now, what step sizes did it take? Surely only 1 or maybe 2 nanometer steps.
You'd think so, but here's the jaw dropper in Figure 5D. Even with only 2 active subunits, this RWE meter, it still took steps ranging from 1 nanometer all the way up to 4 nanometers.
Hold on. It made 4 nanometer steps with only 2 working engines.
Mm-hmm.
Where does the energy for the other 2 or 3 implicit steps come from if those subunits can't burn fuel?
That's the million-dollar question it answers. It implies that large physical steps do not require simultaneous or sequential ATP hydrolysis in all the participating subunits.
So what's the model then?
Yep. The idea is that one of the active W subunits fires stochastically, it burns its ATP. That single event initiates the power stroke.
Okay, that gives you maybe 1 nanometer?
Maybe. But that initial mechanical action triggers a rapid, coordinated cascade around the ring. The structural change propagates. And the energy for the subsequent parts of that large step might actually come from the release of ADP or even ATP that didn't get hydrolyzed from the neighboring inactive subunits.
So the dud subunits still contribute by just letting go of their bound nucleotide when pushed by the first one.
That seems to be the case. The mechanical wave forces them to change shape and release their nucleotide, and that release itself generates force, contributing to the overall multi-nanometer step. It's like coordinated energy release, not just coordinated hydrolysis.
Wow, so it's using latent energy or the energy of binding release from the inactive sites to complete the big step started by just 1 or 2 active sites.
It seems so. Stochastic initiation followed by a coordinated completion cascade.
That's a fundamentally different way of thinking about mechanochemical coupling in these motors.
It absolutely is. It moves us way beyond simple, rigid models.
Okay, so we have this incredibly detailed picture from the tweezers. How does this relate back to what happens in, a real cell environment? Is this unfolding and translocation the slow part of protein degradation overall?
Great question. Figure 6 looks at that. They compared the time the motor spends actually unfolding and translocating, what we just talked about, plus $, with the total time it takes for a protein to get degraded in a test tube, sort of mimicking the cell.
This is pulling the slow part?
Nope. The actual mechanical work, the pulling and spooling, was way faster than the total degradation time.
Really? So the motor itself is fast once it gets going?
Blazing fast, relatively speaking. The real bottleneck, the slow step in the overall process, seems to be what they call the commitment time.
Commitment time?
Yeah, that's all the stuff that happens before the pulling starts. The enzyme has to find the right protein, bind to it, thread that little tag into the motor's opening, and really lock on and commit to unfolding it.
So the setup takes longer than the execution.
Often, yes. For some proteins, this initial engagement phase can take like 4 times longer than the actual unfolding and translocation part. Finding the target and getting a proper grip is often the hardest part for the cell's shredder.
That's a crucial takeaway for you listening. A protein's fate isn't just about how fast the motor pulls. It's about 2 things. First, how stable is that protein right near the tag? That determines if the shredder can even start pulling it apart effectively. And second, there's this whole complex dance before the pulling even starts finding and engaging the target. That's often the real rate-limiting step.
Exactly. And then figure 7 kind of puts it all together in a final model. You have ATP binding, then a stochastic probabilistic hydrolysis event in one subunit.
That sets the size of the step, 1 to 4 nanometers.
Right. And that initial firing kicks off a rapid, coordinated cascade of nucleotide release or maybe further hydrolysis events in the neighbors counterclockwise to complete that multi-nanometer burst. Stochastic initiation, coordinated completion.
It's a beautiful quantitative picture. It really gives us the grammar of how this machine steps.
It does, and it moves the field forward from these kind of vague, averaged out results to seeing the actual nanomechanics at play.
And presumably, this mechanism, this blend of randomness and coordination, might apply to other similar AAA+ machines in the cell. There are lots of them doing important jobs.
Very likely. This idea of stochastic initiation coupled with coordinated cascades could be a general principle for how these complex molecular machines work. It might even give us new ideas for, say, antibiotics, maybe drugs that don't just block the fuel burning, but specifically mess up the coordination between subunits.
Fascinating. So, okay, final thought then. We've seen ClpXP can take these huge steps, potentially getting energy just from nucleotide release from subunits that can't even burn fuel themselves.
Yeah, essentially recycling or leveraging binding energy.
So the provocative thought for you to take away is, what other hidden energy sources or non-obvious mechanisms might other molecular machines be using? Are we maybe underestimating how cleverly they use structural changes in nucleotide binding or release, not just hydrolysis, to get work done?
It definitely makes you wonder if there's more subtle energy management going on in these nanomachines than we currently appreciate. A lot left to explore.
Absolutely. A fantastic deep dive into the mechanics of life. Thanks for joining us.